I'm here to speak to you about the Zava-Dos of biology so you can pass that AP Bio exam. So Ryan, what Zava-Do we remember about cells?
Well, Jamie, you see there are two different kinds of cells. You have plant cells over here and animal cells over there, and they have some differences, you see. Like plant cells, they have cell walls and chloroplasts and central vacuoles.
So what Zava-Do we remember about plant cells?
Well, really the only difference about plant cells is that they have a cell wall, chloroplasts, and a central vacuole. The cell wall is basically what, you know how you bite into a piece of lettuce and it's crunchy. That's the cell wall. It makes it crunchy. It's the protection. And also, the chloroplasts are where photosynthesis occurs and where they produce their own food and how they produce oxygen for us to breathe. And then there's also the central vacuole, which contains all the water of the plant, and it produces something called turgor pressure, which makes plants able to stand up straight. It gives them their stance. It gives them their posture, basically. It allows them to not just fall over.
So Tony, what does photosynthesis look like?
Well, if you look over here,
Photosynthesis.
Photosynthesis. Photosynthesis. Photosynthesis.
Photosynthesis. He's nuts.
and that's what photosynthesis looks like.
So Tony, what Zava-Do we remember about cell communication?
Well, the thing about cell communication is that in animal cells, they have things called gap junctions, which allow communication between each animal cell. But in plant cells, they have things called plasmodesmata, which are basically holes in the cell wall that allow the communication between each plant cell. So there are differences, but in a way they're the same.
So Jamie, what Zava-Do we remember about certain cell membrane components?
Well, we Zava-Do remember that certain proteins act as transporters, carrying what goes inside and outside of the cell. Some act as enzymes, carrying out chemical reactions on top of the membrane. Some act as surface receptors, receiving signals from other cells. And they also act as identity markers, kind of tagging the cell and telling other cells what function it has.
So Jamie, what Zava-Do we remember about diffusion?
Well, we remember that solutes like to diffuse from an area of high concentration to low concentration to find equal the concentrations out. And we can also remember about facilitated diffusion, where a membrane protein tries and helps push all those solutes through the membrane and make it an easier process.
There is also a thing called osmosis, which is just basically the diffusion of water. And water, as we know, is a universal solvent, and many solids dissolve in water. But there's also one thing we have to remember about osmosis. Water always diffuses down the concentration gradient.
So Tony, what Zava-Do we know about active transport and bulk transport?
Well, the thing about active transport is that it allows cells to take in substances or molecules that already have a high concentration in their extracellular fluid. And also, bulk transport is a process called endocytosis, which includes phagocytosis and pinocytosis. And phagocytosis is basically when a cell just takes in an organic matter or solid. And pinocytosis is just basically when a cell takes in a fluid.
So Tony, what are some Zava-Dos on cell communication and reception?
Well, the receptors are on the cell membrane, and these receptors have specific signals that they receive, and these signals are called ligands. But also, the receptor's location on the membrane defines what type of signals they receive. And also, that's it.
So Ryan, so Taylor, what are the Zava-Dos about adaptive proteins?
Oh, you see?
Oh.
Yuck. Adaptive proteins act as a link between the receptor and proteins that initiate downstream signaling events. For example, the RAS protein. Oh my god!
Oh, zippity-doo!
Discussed later. Is activated by adaptive proteins
Binding to a receptor. Oh my god! Zava-Do!
This is an example of mitotic cell division.
Mitotic's a word. Stop.
As you can see, in the middle, represented by the tape, is the cleavage furrow, where the cells will eventually divide and create two identical daughter cells.
She likes to do it.
And here's the rest.
This is when we're starting to break apart. You see, we're in like the last phase of
They will soon split.
Will we? And now we're two cells. Well, Tony, what are the Zava-Dos of bacterial cell division?
Well, bacterial cells go through a thing called binary fission, which is just asexual reproduction and produces two copies of themselves, since they don't have a true nucleus like a regular eukaryotic cell.
No brain. So, Jamie, what are these Zava-Dos of karyotypes?
Well, a karyotype is the 23 matched pairs of chromosomes, also known as tetrads, in order from largest to smallest, telling them, pretty much mapping them, showing them their characteristics and traits. So, Tony, what are some Zava-Dos about mitosis?
So, in mitosis, there are things called, there's this interphase, which has 3 steps to it. That's G1, S, and G2. Those are the 3 steps to interphase. And in the G1 phase, that's actually where most of the growth of the cell happens in preparation for the cell division, since mitosis produces 2 identical daughter cells. So in the S phase is where the chromosomes replicate to produce 2 sister chromatids, and they attach at their centromere and they form a tetrad. And also there's the S2 phase, G2 phase, where just the chromosomes just coil more tightly.
There you have
So, Jamie, what are the Zava-Dos about the M phase of mitosis?
Well, the M phase is when you have a bunch of condensed chromosomes and they attach themselves to the mitotic spindles. And then those spindles start pulling to the opposite sides of the cell, creating almost 2 different cells. And then you get a cleavage furrow in the middle that starts to break apart the cell, making it 2 identical sister cells.
And the cleavage furrow is what part? Wasn't it the cytokinesis?
Exactly, the cytokinesis.
That's what happens during cytokinesis?
Exactly.
And then it produces the 2 daughter cells, which are identical copies of them.
Absolutely.
How do you
Alright,
get
we'll
to
turn.
cells?
Go first.
Cells.
Oh, no.
Cells. To the back.
Cells, where we get those kill.
Where'd you get those cells? Stop! I'm still attached to it. Oh my god! My head! Ow! Oh my god! What the? You freaking jerked me out!
Vacuole, which is filled with water.
Keep going.
Said that right in the middle of the thing.
Keep going.
Not be a dumbass.
Keep going. What? I was zooming in on him.
Okay.
Yeah, we can't use that after what I just did.
Completely hate water, and that's why they're inside. But that's
Location dependent.
in communication.
Well. I'm gonna keep a straight face.
Well, I'm really
Quail.
Just read about scaffolding.
No, adaptive proteins. Yeah.
Are you not? Wait, no. Do scaffolding proteins because we actually learned about that.
I don't remember that.
How do you not remember scaffolding proteins?
You have the book right in
Here's
front
the
of you.
definition.
We have these guys right here. I'm gonna ask him about these guys.
Adaptive proteins.
Okay, I don't give a crap anymore. I just want.
Can I twerk over there? Can I twerk over there?
Okay, go.
So Ryan.
So Tyler.
Is when the sister chromatids. Is when this.
Make it to the camera. We're going to the camera. Oh my god! What are we supposed to be saying? I don't remember. What are we doing?
Can you not record this?